Have you ever wondered what actually happens before Bayer launches a new drug onto the market? Or thought about all the things that have to be taken into account in the process? Let's look back over about 12 years. Everything begins with Bayer determining a high unmet medical need in a certain therapeutic area and deciding to search for an active substance that will help the patients. The research begins by looking for a target, like a physiological mechanism, that can be addressed by a drug. To do this, the researchers must have precise knowledge of the biochemical processes caused by a disease in the body. Great care must be taken here because the quality of the target will later influence the success of all the other work steps. The next stage is high-throughput screening. You might say it's like looking for a needle in a haystack. The scientists use a systematic testing procedure to search for substances that could be a suitable starting point for a new drug candidate by screening about 4 million chemical compounds. For this purpose, Bayer provides them with one of the biggest compound libraries in the world. Up to 400,000 substances can be tested per day. At this point, it's still impossible to say which of these substances will be the starting point for a later drug candidate. This is because the remaining candidates can be compared with key blanks. You can insert them into a lock, but they wouldn't open the door. Medicinal chemistry is needed to give them the finishing touches. So Bayer's scientists compile hundreds or even thousands of different variations. The candidates that best match the requirements make it to the next round. The next step is carried out by Bayer's pharmacologists, who study what the substance does with the body under many different experimental conditions. They check whether it really can treat the disease. And at the same time, the pharmacokinetics experts examine what the body does with the substance. Does it even reach its destination? Or is it, for example, broken down in the liver before it can even get there? Parallel to this, the toxicologists examine potential side effects and determine the therapeutic window. That is, the dosage range in which the substance has a therapeutic effect without having unacceptable side effects. But computer simulations and test tube experiments are not enough to generate reliable findings. Animal experiments are vital to understanding the complex interactions that take place in an entire organism. These experiments are required by law and governed by strict guidelines and state controls. 5 years have already passed by now. Thousands of substances have been synthesized, tested, and further optimized on the way here, until a single development candidate has ultimately been found that meets all the necessary requirements. But there is still a long way to go before the drug is approved. Before starting the clinical test phase, the process will only continue if the candidate complies with this development level's strict criteria on efficacy, safety, and developability, and if all the departments involved give the green light. Parallel to this, tests are already being conducted on how the drug can later be administered. For example, as a tablet, an ointment, or drops. Furthermore, Bayer must make sure that it can be manufactured not only in the laboratory, but also on a large scale at a consistently high quality level. Its storage safety and durability are also thoroughly analyzed. This is where the most exciting phase begins for all the people involved. The move from the laboratory to clinical trials, where the drug's safety, tolerability, and behavior in the human body are tested on people for the first time. Many new developments don't even make it this far and are discontinued beforehand. Before a clinical trial can begin, it must be approved by the regulatory authorities and an independent ethics commission. The study participants are also informed in detail in advance and must officially give their consent to taking part. In Phase 1 of clinical trials, the development candidate is usually tested on small groups of healthy volunteers. The main purpose here is to determine the tolerability of the active substance, to find a safe dosage range, and to identify possible side effects. Interactions with foodstuffs may also be investigated. Only if Phase 1 is successful can the next phase begin. Phase 2 is one of the decisive stages in drug development. It is where the suitable dose is determined and the efficacy and safety of the drugs in a specific indication are studied with the help of a small group of patients. The findings indicate, for the first time, whether the drug really does have the expected effect in the patient's body. If Phase 2's results are also positive, it is followed by Phase 3, involving often thousands of patients worldwide on average. This is where the decisive facts and data are collected for the benefit-risk assessment, and the efficacy and safety are thoroughly investigated and confirmed in larger groups of patients. This part of clinical development involves a very complex and lengthy process. Over half of the total cost of developing a new drug, which averages more than €1 billion, goes into these studies. If Phase 3 is successfully completed, the new drug is submitted for approval. For the approval process, Bayer compiles a dossier containing all the data collected during development. All in all, it contains a total of more than 500,000 pages. If they were all printed out and placed on top of each other, the stack would be almost 50 meters high. The approval authorities now decide whether sufficient evidence has been submitted to prove the drug's efficacy, safety, and quality for the proposed indication. They weigh up whether the benefits outweigh the possible risks. This examination process takes from 1 to 2 years, after which the drug has finally made it. The official seal of the supervisory authorities confirms the product's approval for the market. Even after approval of a product, Bayer's medical experts continuously monitor its benefit-risk profile according to legal requirements, health authority regulations, and internal guidelines. Reports of side effects or adverse reactions are collected and analyzed by our teams. These analyses are consolidated with the existing benefit-risk assessment and shared with health authorities. If necessary, new findings or steps to further mitigate risks for patients or users of our medical products are determined. These can include specific recommendations for use, warnings, or restrictions of the patient group in the product label or package insert. Many side effects are detected during drug development, but very rare side effects are sometimes only found after the product is on the market, when more patients get access to it. That's why Bayer continues to collect healthcare data in extensive studies under real-life practice circumstances. While the newly developed product is helping patients all over the world, Bayer's scientists have long since begun researching new drugs, synthesizing, optimizing, testing and combining substances, thus contributing to medical progress. The focus in this context is always on the therapeutic benefit for the patient.
Bayer. Science for a better life.